Today's talk is titled Identifying Real Changes in Islets and Beta Cells During Progression to Type 1 Diabetes. And Dr. Clayton Mathews is Professor and Chair of Infectious Diseases and Immunology at the University of Florida. He's going to present. Despite continuing improvements in the ability to treat individuals with type 1 diabetes, the need to identify a true cure for the disease remains. You got that right. To meet this need, the Mathews Group seeks to improve their collective understanding on how T1D develops in humans by leveraging blood samples from research volunteers, human tissue samples, and preclinical mouse models. The overall research done in the Mathews lab has been to identify the processes that result in the clinical symptoms of T1D by focusing on islet physiology, immune effectors, and genetic loci that encode risk or resistance to T1D. And the current project has used human tissue samples to study function, gene expression, and proteomics in non-diabetic, autoantibody positive at risk or recent onset T1D cases. And the lecture is going to relay their observations and elaborate how beta cells change prior to diagnosis. I'm going to drop the paper, the link to the paper into the chat, and please drop questions here during the into the chat during the talk, and Dr. Mathews will take them as they come in. And hold on, just admitting the rest. Thank you so much. We're really, really interested in this and sort of the role of the delta cells in what you're seeing. So take it away.
Thanks, Monica, and I hope everybody's doing well. So this is a project that has been brewing for quite a while, probably a dozen years or so. It has finally begun to result in a few papers. What have I done wrong here? Here we go. The one that we are, I was requested to talk on today, which was recently published in Diabetologia. So running collaboration with Wei-Jun Chen and Rohit Kulkarni on looking at proteomic changes in the islet. Then have a paper recently accepted for publication, which is really closely tied to this proteomics paper. And that paper was published by Molly Huber, who's now a postdoc with Rohit Kulkarni. And that project was in close collaboration with Ed Phelps Group here at the University of Florida. And then we have another study looking at the gene expression profiles in islets across the 4 groups: non-diabetic, single antibody positive, multiple antibody positives, and those with type 1 diabetes, but insulin positive beta cells.
Yeah, we've had a bunch of University of Florida talks recently, a lot of exciting papers coming from University of Florida right now.
So the whole idea here is for us to understand how beta cells change towards diagnosis, so we can get a better handle on what would be the right way to treat these cells. I think over the past 15 or so years, I think we've begun to understand that there are 2 major failures in type 1 diabetes. There's the failure of the immune system that's likely regulating and controlling death of the beta cells, but there's also failure of the beta cells. And if we don't address beta cell failure and insulin production, then we don't have a great way to cure type 1 diabetes. So I'll start by acknowledging this is a massive collaboration. As I mentioned, Ed and Wei-Jun and Rohit, as well as Martha Campbell-Thompson and even Gerling, who've all been major contributors to all the work that we'll do today, as well as folks in their groups and mine. Certainly thanks to Breakthrough T1D, JDRF, as well as NPOD. We cannot do these studies without NPOD and the people that are on 24/7 call to work with OPOs, as well as the families of the donors, to get the tissues into our hands so we can understand how type 1 diabetes starts. And thanks to the funding agencies that allowed us to get the work done. So as the title says, our goal is to understand real changes that occur in beta cells. We've had, over the past 40 or so years, there have been lots of work done with isolated islets, treating isolated islets in various ways, either putting immune cells at them or immune effectors. I don't know that those studies have had a tremendous impact on our ability to cure the disease. And so our work over the past 12 to 15 years in trying to identify how the beta cell changes are really based on rationale for studies that occurred probably starting in the 1960s and occurring today that have looked at beta cell dysfunction in vivo in people, as well as beta cell mass and function in vitro based on some recent studies with live tissue slices. So what's appreciated is that there's a decline in beta cell function, specifically first phase insulin secretion in progressors as they move towards diagnosis. This has been published a number of times. This is a very nice longitudinal study that was published about 9 years ago, which shows that there will be, prior to diagnosis, there's a decline in function, which occurs over a year. So it's a relatively slow decline in function, I think, which probably fits very well with the model. This is a slowly progressing disease in a chunk of the individuals that develop disease. This wouldn't include those that are developing diabetes, the youngest that are the really high risk DR3/4 donors. But so while we have this decline in beta cell mass, what the field has not been able to do is associate this with a decline in beta cell mass or beta cell area in antibody positive donors. So a few years ago, there were 3 papers that were between 2015 and 2016 that all had the same conclusion, that there's no decline in beta cell mass in these individuals that are at risk for developing diabetes based on their HLA and their antibody status. So just some preliminary data and some things to think about as we go through the data. From the recent paper with Molly Huber, what Molly was able to demonstrate was that there is, as we would expect, a decline in what we're looking at here is calcium flux in the type 1 islets compared to autoantibody positives and non-diabetics. And this is the calcium flux with high glucose versus low glucose. So as we'd expect, on a per beta cell basis, there's less calcium flux in those with type 1 diabetes, and that the fraction of responding beta cells is reduced in type 1 diabetes as well. But again, these are insulin-containing beta cells. They are failing to respond to glucose by fluxing calcium and therefore secreting insulin. The one, I think, major finding from Molly's paper, and she presented on the Sugar Science a couple of months ago, this paper and these results, was that in our hands, we do not see that the presence of immune infiltration is associated with beta cell failure, in that beta cells in islets that have immune cells and beta cells in islets that do not have immune infiltration have equal dysfunction. And a final piece, and this is something to think about if we try to cure the disease. We've done a great job of identifying and running clinical trials and looking at immunotherapy and the ability to preserve beta cell mass. But if we look across multiple therapies, either TZIELD or anti-CD20 or anti-CD2 or ATG, we see that no matter how we treat these individuals, there's the decline in beta cell function over the 2 years after therapy was initiated. So these therapies may be having an effect on the immune system, but they are not having an effect to improve beta cell function in these individuals with type 1 diabetes. So our goal is to find these changes. What we know is that there's a decline in beta cell function that it begins years before diagnosis. We are not observing a decline in beta cell mass or area. We do see that beta cells in cases with type 1 diabetes that have insulin, they are dysfunctional, and that's independent of immune infiltration. And that when we treat individuals with immunotherapy, there's not great rescue of beta cell function. So we're impacting potentially the immune system, but we are not really treating the beta cell in a way that's allowing it to recover function and maintain euglycemia. So I think the goal here, and I think a major goal of the field, is to understand how and why the beta cells fail and how we can intervene to reduce that dysfunction. So for the paper that we have just published this month in Diabetologia, Adam Swenson is the first author. It was co-first authored across the groups that were involved. We've used global proteomics with pooled islets that we've captured using laser microdissection from stage 1 type 1 diabetes cases. And we'll get to the cases in just a second. But we pooled islets, 200 islets per case, and we had 2 replicates per donor. And those were run through a platform that would allow minimal sample loss, and then down to the mass spec that allow us to get about 4,500 proteins per case. For this study, which started about 10 years ago, maybe 11 years ago, we had a total of 10 in-pod cases, 5 with antibodies, and 5 that did not have autoantibodies. The block above of the 6, which are the 6, the 3 multiple autoantibody positives and their age and sex matched controls, are the ones that we followed through. We did have a protective HLA in one of our antibody positive cases, 6044. But when we clustered the proteomes of the cases, what we saw was that the single autoantibody positives were quite different than the multiple autoantibody positives. And I think we were able to make excellent progress in analyzing the multiple antibody positives to their age and sex match controls. And this just shows some QA/QC that we do have the ability to differentiate the autoantibody positives from the controls. So from a standpoint of whole islet proteome changes, and this is untargeted proteomics that we're studying, we had a few major findings. I think as you guys could imagine, and perhaps not going in order as I should, but I should have put these in order. But we do see, and part D would be, we do see an increased inflammation. But I think there are some quite interesting findings here. And one of the major ones is that we do see a reduction in the machinery for translation of RNA to protein. We saw an increase in some pathways for glucose metabolism. These would be notably an increase in GAPDH, which is a member of both glycolysis and gluconeogenesis. I think surprisingly, and we'll get into this more later with 2 of the other studies we have going on, we do see a global reduction in an ER stress response. And I know that the field has really worked on ER stress, and there are a lot of people in type 1 diabetes that think beta cells die by ER stress. But to date, I don't think there's been a strong case made with human tissue that ER stress is a major player in type 1 diabetes. And at least the 3 studies that I'll talk about today, we do not see that ER stress is a major player. It's either reduced or not different across the groups with either prediabetes or diabetes. And that could be the fact that if there's this reduction in global protein production, that there would not be a need for ER stress if the protein load is lower. So we have, and so sort of shifting from paper 1, where we've identified these global changes, in sequence, part of what Molly was doing was taking the data we had from Adam's paper, and then looking at parts of the glucose-induced insulin secretory machinery on a per beta cell basis. So Molly would have presented these data a couple of months ago, but this is a recap of what she has seen. And when we looked across glycolysis as well as mitochondrial function, what we saw was a decline in the ability to transport glucose, metabolize glucose, although GAPDH is elevated, perhaps more so in the nucleus, and we could probably talk more about that in the question section. We also saw a decrease in electron transport and oxphos subunits. And so when we looked at these, we see that insulin was not different by protein on a per beta cell basis. We do see, as expected, there's an increase in HLA in the type 1 diabetes group. We did see a decline in GLUT1. in members of glycolysis. We do see this upregulation in GAPDH, and then succinate dehydrogenase C, which is a subunit of succinate dehydrogenase, which is a member of the TCA cycle, as well as the electron transport chain is reduced in both multiple antibody positives and T1Ds. And we can see that some of these are verified at the RNA level. So in global, what we saw from taking the data from Swinson and then looking at subsets of those in Molly's paper was that we do see this increase in HLA. We don't see a change in insulin. We see no change in ER stress, at least for PERK and IRE1 alpha. It's not different. We do see some of the glycolysis proteins changing, potentially meaning that the beta cell is becoming blind to glucose if it cannot transport that glucose in. And then further downstream from glycolysis, the ability to further metabolize pyruvate and generate ATP is reduced in the antibody positives and the T1Ds. So to increase our rigor, we've also looked at this by untargeted gene expression analysis. In this case, we're going to utilize the same four groups: the non-diabetics, the single, the multiple, and the T1D group. In the case of the gene expression analysis, what we've done is we've used immunofluorescence to guide our studies. So if we take a section, we're going to stain the first section with insulin, with glucokinase, with glucokinase, with ATPase, with CD3, and also for class 1 HLA. And then we're able to use these different parameters to do a laser capture microdissection across the full islet, across multiple sections, and then run an RNA analysis on a per islet basis. So as you can see, we can stain for insulin and CD3. And then, as mentioned before, we had 10 donors in the no diabetes group. There were 4 single autoantibody positives. And by staining for the current study, we're really going to focus only on the CD3 negative islets. But we had 2 islets in the single antibody positive group that had insulitis by the definition, 6 or greater immune cells. The multiple antibody positive, we have 18 islets without insulitis and 7 with, and then the type 1 diabetes group, which we would have far more insulitis. So in the studies we're going to talk about now over the next couple of minutes, we're really going to focus just on the CD3 negative insulin positive islets, because we want to know what's occurring in beta cells in the absence of overt inflammation. So in that case, we do see that there are lots of genes changing if we compare singles and multiples or T1Ds to the non-diabetic group, right? Roughly 1,000. And we're seeing that the bulk of these genes are being suppressed in their expression. If we compare the islets, and now we want to know what's similar across the 3 groups of islets, from the singles, the multiples, and the T1Ds compared to the non-diabetics, then we can take what is several thousand genes and shrink that down to maybe 2,500 genes to start to make conclusions on how beta cells are changing during the process in the absence of inflammation. So similar to what we had seen with the proteomics, we saw a large decrease in the ability of the islets in the single, multiple, and T1D groups in translation initiation, regulators of translation, as well as ribosomal proteins, but large and small. We see a decrease in proteins in the ER, as well as the ER stress response. And we see a decrease in lysosomes, which would suggest also a decrease in protein turnover. getting back to beta cell and glucose metabolism and this idea that beta cells are going blind to glucose during progression to diabetes. We overall are seeing a decline in enzymes for glycolysis, for TCA and mitochondrial metabolism, in mitocarriers for solutes, including the ANT translocases, as well as subunits across multiple complexes for electron transport. So I think overall, the three studies are telling us that in type 1 diabetes, we see this increase in immune response genes. But if we look back at the single and multiple autoantibody positive cases, we really don't see the increase in immune response genes, but we do see these decreases in protein synthesis, the ER stress response, as well as the metabolic response. When we've done immunofluorescence to confirm those, we see that, again, we can confirm that the ER stress response is down and that these metabolic processes are down. And then when we further confirm that using gene expression analysis, we again see there's a reduction in protein synthesis, ER stress response, and metabolic response, specifically glycolytic and mitochondrial response. That certainly would have the potential to cause a decline in glucose metabolism and insulin secretion. So overall, we see this decline in glycolysis. We do see the elevation in GAPDH, and that's something that's interesting from perhaps a stress response standpoint. We see a decline in the TCA cycle, likely leading to a decline in ATP. In the antibody positives, we're not yet seeing a decrease in calcium response, and it's likely that there's not quite yet the buildup in the decline in these proteins where we've hit a critical mass that they can reduce ATP and calcium flux. But once we get to progression, what we see is that there's an inability to flux calcium and an inability to secrete insulin in these islets, even though they do have insulin. So with that, I will stop and answer questions.
Thank you so much, Clay. If anyone would like to ask a question, please either raise your hand or just unmute yourself. I have a question. I mean, how can you kind of dissect whether or not, like, could it initially be the mitochondrial dysfunction that sort of drives the decline and alteration of the glycolysis? Or, are they both going offline? Is glycolysis going offline first? What are your thoughts around that?
So it might be easiest to think first if there's this decline in proteins translation, then we would see potentially a global decline across protein production, which may also lead to a reduction in the production of mitochondrial subunits in general, specifically those that are nuclear encoded. So if we're seeing a overall decline in protein production, we're seeing an overall decline in mitochondrial mass, and that's then resulting in a decline in the ability to produce ATP for first phase insulin production, which is that major phenotype for progressors to type 1 diabetes.
Well, what percent of mitochondrial proteins are nuclear encoded?
So, okay. So there are hundreds of mitochondrial proteins. Only 16 are encoded on the mitochondrial genome. So the vast majority of the proteins in the mitochondria are nuclear encoded mitochondrial proteins.
Yeah. Yeah, like the human mitochondrial DNA encodes only 13 proteins, right? Those are all like in the OXPHOS system.
Well,
Search.
yes and no. So over the past few years, people have been identifying some novel mitochondrially encoded proteins. Humulin is one that's encoded on the light strain anyway. So the number has increased recently based on these more recently identified proteins, but the majority of those, the 13 proteins, yes, that we think of are commonly expressed from the mitochondrial genome are part of the electron transport or ATP synthase complexes.
Where do those sit? Are they like on the, are they all over
So,
the
yeah,
chromosome? Are they some like fragile sites?
so if you look across the genome for subunits of complex 1, they
Hmm.
would be all over the genome.
Hmm. And so also like how do you kind of come to terms with Sarah Richardson's, exciting discovery of the endocrine objects, the smaller size of these endocrine objects. And she's here on the call, so maybe she could chime in. In those who are heading towards type 1, you know, uh,
We'll
does
ask Sarah to
that,
unmute and
Sarah, can you unmute?
clarify.
Yeah, I mean, does that phenotype kind of dovetail into what you're seeing?
Hear me?
We
Yes,
can.
we
Hi,
do.
Sarah.
Hi, how are you, Clayton?
I'm well. Hope you're
Good.
well.
Yes, good, thank you. Yeah, just I guess following up from what Monica's kind of pointed out. So we're got the data that suggests that we're losing the sort of smaller endocrine objects in type 1 diabetes, but you're looking in the autoantibody positive donors mainly here, and we've got some data kind of coming out where we've started looking in those, and in the single autoantibody positive donors, we don't see loss of those small endocrine objects. And I'm assuming that because you're doing the kind of stamping out of the islets, you're picking the bigger objects to do these studies in anyway.
So I would say no. So for the proteomic study, we were cutting as much as we could, but I do think there's probably some diameter where we weren't cutting those. I will
Yeah,
agree there. From the gene expression studies, because those were capturing the same islet across multiple sections, then
yeah.
we would have captured, the tip of the tip of the islet all the way through the wider section. Um,
I
so
guess what we're talking about when we're talking about the small endocrine objects are literally the ones that are single
yeah,
cells or very small clusters of cells. And I suspect these are probably not being picked up in the studies that you're presenting here.
that's correct. So we would, if we went back in the studies where we've done immunofluorescence then we could ask the question about whether or not the single islets or the clusters of a few beta cells, the single beta cells or the cluster of a few beta cells, do they change in the same way that the larger endocrine objects change?
Yeah. And I
Those
think that would
those
be
data exist, right? Because we have all the images. We could ask that question.
I think that would be interesting to do, but absolutely. I think our data is suggesting that you're not losing them at least in the single GAD autoantibody positive donors. We are starting to see some loss of those small objects in the multiple autoantibody positive donors who've got kind of other signs that they might be progressing.
Hey, and Sarah, I want to ask you another question. This is something that we've struggled with, and David and Joy, I'll get your questions in a second. So there are a lot of single GADs with protective HLA. We have tried as hard as we can. I realize that we did have a protective HLA single in our initial proteomic study. We excluded that for the downstream studies, I think for obvious reasons. Have you been excluding protective HLA donors as much as possible
Um,
from your studies?
We're just going through because we've just expanded the numbers into the single and multiple autoantibody positive donors. So we're just going through and doing all of that kind of analysis at the moment. But we can certainly break it down to look at the impact of protective HLA in the outputs that we're getting. But we haven't done it yet.
Okay, thank you.
Very exciting times on both sides of the Atlantic. So we've got a question here from David Hill. Did you see any changes in proglucagon or somatostatin gene expression pre-type 1 diabetes?
I don't believe we have, David. And I don't think that those were proteins that came out of the proteomics either. We had some data on insulin and proinsulin, as you can imagine, but I do not believe we saw changes in proglucagon or somatostatin in the proteomics. And from the gene standpoint, I do not believe in the antibody positives that we have seen by gene expression that there were changes in glucagon or somatostatin. And then Julia, Molly's paper, we didn't see any differences in function in infiltrating and non-infiltrating islets. Based on the total, I would say based on how we did the analysis and the number of donors, when we start to split out, because we only had 7 donors per group for non-diabetic, single, multiple, and T1D, and all those were the singles, multiples, and T1Ds were all donors that had risk HLA. When we split those out into cases with and without infiltration, we didn't have enough cases to pull apart differences in the levels of the proteins. They seem to be very similar in like succinate dehydrogenase, which was decreased in infiltrated islets, was equally decreased in non-infiltrated islets. Let's see. Yeah, so Guy, great question. So Guy Rutter is asking about the preservation of calcium dynamics in the antibody positives and whether or not this was due to the fact that we didn't see major changes in rate limiting enzymes. And so the answer to your question, guys, yes. So we do not see, we actually did not stain for phosphofructose kinase, which is rate limiting for glycolysis. We see changes in rate limiting for TCA cycle by gene expression, but we'd say if we had the ability to run twice as many donors, we would have seen OGDH and TCA cycle would have been significantly decreased by the protein level. I do think that part of a limitation of our study is that we have always done 16.7 millimolar glucose. And I think it could be if we had dropped the glucose level, we may begin to see differences in multiple autoantibody positive cases that are trending towards having less protein. Because we don't know where the multiple antibody positive cases are in progression, or if they are the small percent that will never progress at all, it's hard to make I would imagine we're going to have heterogeneity in that group based on sort of where they fit on the timeline in progression. And then so do changes, the other I think important point and limitation of studies, do changes in cell composition confound your conclusions with the gene expression changes? So for the type 1 diabetes cases, I think the answer is going to be there are going to be some limitations and challenges there because we will have more and less beta cell mass due to length of duration of disease, as well as potential for destruction. For the single and multiple antibody cases, I don't think we will. So I think the results we have there will reflect a real change that's prior to diagnosis, that's part of pathogenesis, because we really do not see loss of beta cells. We don't see loss of insulin, and we don't see infiltration to a large extent. I think the continual use of anti-CD3 in our studies for both immunofluorescence, but also for future studies for spatial metabolomics or spatial transcriptomics are going to be very critical for us to make sure that we are able to detect balance of endocrine cells and also presence of immune cells.
Yeah, I mean, this really speaks to to the finding that even with teplizumab, the beta cells come back online, but the remission doesn't last, right? Because they're not 100% back from the dead. Can I just ask you, in terms of like replicating this in the dish, like, have you thought about, like how are you guys going to create the sick beta cell? To kind of like further the study?
Okay, so that's a great question, and Anoop, I will get to your question in a second. So hang on. So we have like I had mentioned before, we've been doing this work for over the past dozen years or so, and we have really begun to think about how do we do a model here that's where we can see the realistic change. So we don't wipe all the beta cells out. We don't cause a massive change that causes dedifferentiation. We find a way to stress the beta cell in a way that causes these minor declines in function. You could say someone who's had autoantibody, if we look at that Koskinen study, right, where people have beta cells failing over 7 years, it's going to be hard to replicate that in a dish. So there are some things we've done wrong, and this gets to the question that I'll answer in a second here about sort of how we treat islets in the dish, where I think Anil Bhushan likes to say that we bathe the islets in massive amounts of inflammatory mediators and just expect this to reproduce type 1 diabetes, and it doesn't. There's some real differences in the way the cells signal if you put in a physiological level of an inflammatory mediator versus the super physiological levels that we put in. And I think we need to start to really think hard about where beta cells live, how do they live, what do they need? If there are, stressors, let's say, we could come back to an age-old stressor, which are viral infections. It could be that repeated viral infections and a poor response based on genetics that are present in those with type 1 diabetes. And there are a lot of genes in the antiviral pathway that are risk genes for type 1 diabetes. And if you look back to, there's a very nice paper that looked at seroconversion genes, and a lot of those genes that regulate antiviral pathways are those early genes in type 1 diabetes. It very well could be that these sort of repeated chronic inflammatory situations are going to lead us to these declines in function. So I would say at the moment we are working diligently on how to do that, and I'm not sure yet. Okay, so I want to get at this question that given that ER stress is the most common urban legend in type 1 diabetes.
You inserted it.
So
yeah, Anoop Aranagiri. Anoop, do you want to ask it?
Yeah, I can. Hi, nice talk. So I just was curious because over time when we are looking at the literature, we always see that cytokine treatments kind of trigger ER stress. There's a lot of literature supporting that. But in your system, you clearly show at least and towards the end, I agree that you don't see such phenotype. So I was wondering what's that distinction? Is it because in the in vivo settings you don't have that much of the cytokines exposure, or it's too much that it does something else? So what's your view on that?
So yeah, so long held belief, I think people have been thinking about ER stress and beta cells and type 1 diabetes for about 20 years. I guess even though the tissues have existed, none of the people that have been looking at ER stress have taken INPOD or HPAP and had said, hey, here's ER stress in an antibody positive at-risk beta cell, or here's ER stress in a type 1 diabetes beta cell. I think that this decline in protein production is resulting in less protein in the ER, and it's resulting in no change in ER stress or a reduction in ER stress. One of the things that at least I think about in the other part of my job, which is more chronic inflammation in humans and sepsis, is when we're getting ER stress. So ER stress does something that's interesting, right? So it stops protein production and stops protein from getting to the cell surface. One of the common hallmarks of type 1 diabetes is the upregulation of HLA on the cell surface. And it's well established that ER stress blocks HLA from getting to the cell surface. So to me, there have been these opposing views that ER stress is a major player, but if ER stress blocks class 1 HLA from getting to the cell surface, then why do we see HLA class 1 on the cell surface as a major hallmark of type 1 diabetes? I think that when you treat islets with cytokines, you're treating them with so much that they're doing a number of things. We see, if we begin to titer down the dose of interferon, if you throw 1,000 units of interferon gamma at an islet, you activate every stat in the islet. So stat 1 through 6. If we use 100 units, we activate stat 1, or depending on the cell, in T cells, we activate stat 4. So you get a more canonical signaling when you use probably a dose that's less. So I think that it's critical for us to think about, at least here, right? What I want to present is, this is what we're seeing. There's a lot of follow-up work to be done here from the global proteomics to say, yes, these changes are actually in beta cells, or from the gene expression, yes, the protein products are actually down in beta cells. And we're doing that now. And Molly did a nice job with her study on that. Yep. So I think it's, and as Monica had asked, right? How do we model this? I think it's changing our models in a way that get us to what looks like what's going on in the beta cell in type 1 diabetes. And I don't have a great answer for how we get there yet. I think for us, and I am not absolved from throwing lots of cytokines at beta cells and publishing it. And I'm certainly not absolved of throwing lots of T cells at beta cells and showing that they get killed. And in both cases, we're overwhelming the system with our immune effector. Even from a standpoint of insulitis, we would very rarely see 25 T cells to a beta cell in an insulitic lesion, and that's commonly used in an in vitro model.
Yeah, thanks for the detailed answer. So based on that, I kind of realized that it's more related to in this case, it's related to the protein synthesis decline that's actually bringing ER stress, or less ER stress. Okay.
Yeah.
Okay, thanks.
Sure.
All right. Any more? Last call for questions. Just a really exciting paper. Apologies for the first link, but we've got the right ones in there. So if you haven't read it, you should. And congrats to Molly, Ed Phelps, Clayton Mathews, and the whole group that put these papers out. It's really interesting to start thinking about this in a new way. So thanks again for sharing it with us today. And we look forward to your next steps and what's coming next out of University of Florida. I'm sure it's going to be something exciting. Have a great rest of your day.
Thanks, Monica. Thanks, everybody, for your time.